Morning, everyone. My name is Alisha. Today, the research I gotta talk about is called Hypoxia aggravate the effect of ocean acidification on the physiological energetic of the blue mussel. So first, what is hypoxia? Hypoxia is a reduction in dissolved oxygen level to 2.8 microgram per liter. And hypoxia can attribute to temperature and salinity stratification in the water column, especially in the summer. And here is the hypoxia area. And what is ocean acidification? We can simply say that the ocean acidification is the seawater become more acidic because the increase of atmospheric carbon dioxide dissolve into the water, and it will trigger a change of complex chemical reaction. And what is physiological energetics? Physiological energetics consists of 6 parameters in this research. And the first one is clearance rate. The second one is absorption efficiency. And the third one is respiration rate. The number 4 is ammonium excretion rate. And number 5 is oxygen and nitrogen ratio. And the final one is score for growth. So first, the clearance rate is that calculate the number of suspended particles removed per unit time in 60-minute assay. And the second one is absorption efficiency, in measuring the organic component in the excreta of the mussel. And the third one is respiration rate. It measures the difference in initial and final oxygen concentration for 1 hour by an oxygen meter. The number 4 is ammonium excretion rate. It measures the difference in ammonia concentration between the experimental tank and the blank tank. And number 5 is oxygen and nitrogen ratio. And this is the equation of the oxygen and nitrogen ratio. The value is equal to the oxygen consumption divided the ammonia nitrogen excretion rate. And the last one is score for growth. It's according to the energy balance equation. And this is the energy balance equation. We can see that the AB is equal to absorption rate, and R is equal to the energy loss in respiration, and the U is the energy loss in ammonia excretion. And what is blue mussel, Mytilus edulis? The blue mussel is a common name, and Mytilus edulis is the scientific name. This is a picture of blue mussel, and it's such a valve, and is commonly found on the beaches. And blue mussel are subject to commercial use. Why do we choose blue mussel? Because it's easy to get, and they are vulnerable to environmental stressors, so they are suitable for experiments. And the blue mussel is also selected as model organism to assess the impact of environmental change in coastal system. So why do we want to do this research? Because the ocean acidification and hypoxia is a stressor to marine organisms, especially those in coastal water. And the second reason is that marine bivalves are ecologically and economically important. What do we want to know in this research? We want to know that whether ocean acidification will interact with hypoxia to cause a much stronger effect than their effect in isolation. It means that the effect on their own. And we also want to know whether hypoxia and ocean acidification can cause negative synergistic effect. It means that to cause negative effect mutually on physiology of the coastal organism. So if the hypoxia and ocean acidification can really cause negative effect, the population of coastal organisms would be diminished in the future ocean. And so how to do the research? First, we have 3 replicated tanks, and they're all in 30 liters, and the dissolved oxygen are all in 6 micrograms per liter, and this is the normal condition. And the first one, pH is in 8.1, this is the normal condition, and the second one is in 7.7. This is the intermediate condition, and the last one is in pH is in the 7.3. This is a low condition. And the second one and the last one, they're all in ocean acidification condition. And we will put seawater into each tank and also put 20 mussel into each tank too. Here we have the same equipment in each tank. This is the pH meter to control the pH value. And this one is the oxygen regulator to control the oxygen concentration. And this is a solenoid valve. It can pump carbon dioxide into the water. And their temperature are all in 20 degrees Celsius. And the salinity are all in 25. And here we have another 3 replicated tanks, and the equipment are the same that we just mentioned before. We will also put 20 mussel into each tank, and the only difference is that their dissolved oxygen are in 2 micrograms per liter, and this is the hypoxia condition. And that's we can proceed to the result. We can see that the clearance rate and absorption efficiency are the same. Their value decrease with a decreasing pH and a decreasing dissolved oxygen, and as the time progress. Their lowest value are also in the lowest pH and the lowest dissolved oxygen on the last day. And the respiration rate is the same that we just mentioned before. The value decrease with a decreasing pH and a decreasing dissolved oxygen. And the lowest value is also in the lowest pH and the lowest dissolved oxygen on the last day. But the ammonium excretion rate is in contrast to the parameter that we just mentioned before. The value increase with a decreasing pH and a decreasing dissolved oxygen as the time progresses. And here is the oxygen and nitrogen ratio and the sulfur growth. They are the same that we just mentioned before. The value also decrease with a decreasing pH and a decreasing dissolved oxygen. And the lowest value are also the same. The lowest value is in the lowest pH and the lowest dissolved oxygen. And let's look at this one. This is a negative value because of the energy loss in ammonium excretion and the energy loss in respiration are too much, so it will lead to a negative value. And the general result, we can see that the time and dissolved oxygen and pH significantly affect all parameters of the blue mussel. And there are also significant interactions between dissolved oxygen and pH, between dissolved oxygen and time, and also between pH and time. And the following is a discussion and conclusion. We can see that the results suggest that blue mussel may be negatively impacted by combined acidification and hypoxia. And there's also the effect of low dissolved oxygen was most prominent under low pH condition in the latter day. It means that the low dissolved oxygen will be more stronger than when it under the low pH condition. And what are the impacts on the environment and the society? The environment impact is that the population of the blue mussel may diminish. And because the blue mussel acts as a food item for many predators in coastal ecosystem. So if the population of the blue mussel really diminish, the coastal ecosystem may be disrupted. And in the society aspect, that the yield may be reduced and the quality mussel may be lesser for fisheries and aquaculture. And this is the contribution of the research. We can see that the data are useful in nearshore and estuarine variability, where blue mussel often reside and where shellfish aquaculture is most intensive. And the contribution is that it is good for economy. And the final one is future outlook. The author think that the more transgenerational study are needed to find the long-term impact of global change on mussel physiology. Peter.
Could you tell me, could you compare the pH value graphic? And I want to know why did you decrease? I'm so curious. Do you see what I mean? I want to know why did you decrease?
Decrease what? Which page?
Yeah.
Could you please repeat again?
I want to know the oxygen and ratio and score for good decrease.
Decrease.
Yeah.
Why? Okay, repeat. You want to know why oxygen and nitrogen ratio and score for growth decrease? Because the oxygen and nitrogen ratios equation is oxygen divides nitrogen equation, and because in the severe condition the nitrogen will discharge more from the blue muscle, and the score for growth is such like the blue muscle is healthy or not. So if the this one here, and the R and U are too much, so it will decrease. June.
And I want to know how to control oxygen.
Oxygen. She wanna know how to control oxygen. This is an oxygen regulator, and it will put air into each tank and to control it. Arlin. The result.
Why is ammonium excretion rate increased?
She want to know why the ammonium excretion rate increase. This isn't mentioned in the paper, but I guess because the blue muscle is put in a severe condition, so it don't have much energy to produce the protein, so it will discharge more ammonia.
Ivan.
So how do you measure the excretion rate? Excretion rate.
Do you hear him? Louder, please.
How to measure the excretion rate?
This one?
Yeah, I guess.
Repeat. How to measure? Just say this one, right?
Yeah.
The method to measure it is using the spectrophotometry. And it's we'll measure the concentration of ammonia in each sample, and it will by the absorption of blue indophenol color, like I don't know how to say it. Yes, it's use this equipment to measure.
Camilla.
Please could you explain why you chose blue muscle that page? Could you explain the environmental stresses?
This
The second one.
and problems.
Could you explain the environmental stresses?
You want to know what is the environmental stressor? In this paper, is mean the hypoxia and ocean acidification.
Ethan.
Can you finish the conclusion?
This one. that the effect of low dissolved oxygen will more stronger when it under the low pH condition. It will affect neutrally like that.
So why do they use
Because the experiment.
Do they have a number to say about this number?
These
Because this is
pictures.
just a chart.
Yes. The paper doesn't give the data. It just gives the picture like that.
Object James, please.
What is source of blue mussel?
Oh, he want to know what is the source of blue mussel. We will take the adult mussel and shell length is 6 centimeters and it collected from here in the East China Sea.
Okay, Jamie, please.
I want to know what's the size used in this study, used in this experiment. What's the
Size.
muscle size?
Size here. You mean this?
Repeat her question.
She want to know what is the size of the muscle, and the size is they take the shell length in 6 centimeters.
So I think you will agree that Alisha gave a very fluent talk, and most of the slides were very clear. But at the beginning there was already a pronunciation. I was okay. How do you pronounce this? No, it's not micro. It's milli. It's 1,000 times difference. It's actually a common mistake for Chinese students. This one is micro. This one is milli. Right? So milligram. And then I think the part of these two environmental conditions, they are nicely presented with figure. But here I think there's some space for improvement. For example, I don't think you need to read through the 6 points here, since you are going to explain the detail here. So if I were you, I will skip these slides and then I will try to see, because these bioenergetic parameters, they are actually not that easy to grasp. It's not that easy for us to understand why this is that. For example, I really have no idea why absorption efficiency is used in SFG. So if you could next time explain either with images or you can draw some analog with daily life. For example, this scope for growth is actually a little bit like how much energy is available for you, for the muscle to grow fatter, or there are not so much energy for them to grow in their biomass. Use easier words, because I think these are probably the text from the paper. All right. If you think you understand, I think you understand. So, if possible, try to use easier words in your own words with the images, with analogs, to explain what they are really about. It's more easier to understand and for us to follow. Otherwise, after you read through the 2 slides and go to the results, we already forgot what they are about. And then the purpose slide is very, very nice. Like this one is motivation slide, motivation, and then this purpose, they are very clear, so these are nice. But when it goes to result, I have the feeling of this part is also nice. But when it came to result, I have the feeling that things went through really quickly. I would actually use the first figure, make it really large, and then you forgot to introduce the axis. You just say, oh, it's declining. Oh, okay, what is declining? And then there are actually 2 bars, light gray and dark gray bars. They are actually different dissolved oxygen. I suggest that next time you take time to explain them well. This actually corresponds to Ethan's question. And I will also notice that because this paper, the result is actually quite straightforward. And here you talk about the effect of low DO under low pH, most prominent in the latter sampling days. I'm not sure if I have missed that. Is there any, all right, okay. Because you did not introduce the horizontal axis, I have been thinking where was the time information? So it's here. So that's something you should introduce. Otherwise, I was just looking at the figure here, which is pH value, right? And then also, I think in this paper, one of the focus is to say that the combined situation of ocean acidification and low DO will make things worse, right? That's one of the points they want to sell. So please show us where to look at that. And when saying that, you can probably tell us what are the stars here. I'm not sure. So what's the CC and the star? Which one means statistically significant? You have the statistics? Or? 统计. All right. Usually this one will mean statistically significant. Others without the star means not significant. So if possible, you can briefly tell us that. And draw our attention to, it's not just this one, but also the combined result. It's actually not, okay, here is more clear. Here, we have the combined result of low DO and low pH. And you see it's clear. The difference is quite large. That means if you have DO and you have low pH, things get worse. So try to draw our attention to that, not only the decreasing trend, because I think it's already a lot of studies showing that decreasing DO, decreasing pH, but you want to show that the combined situation gets things worse. And then also tell us about time. All right. Okay, but otherwise, I think this study has a very, the result is straightforward. So the discussion is also quite straightforward. So next time, just pay attention that especially this conclusion is fully supported by the data that you presented. All right? Okay, it's a good talk. Let's thank Alisha.